The objective for the experiment today is to prepare onion root tip slides and to identify stages of mitosis. Apparatus and materials.
Assalamualaikum and salam sejahtera. Today we are going to conduct experiment cell division mitosis. The first part we are going to introduce you the apparatus and the materials that we need to do for this experiment. First, forceps, blades, dropper, wash glass, mountain needle, towel, labeling, slide and cover slip, thermometer, beaker, tissue paper and filter paper. Okay, for the materials, we need acetic alcohol, 1 molar HCl, acetylocene, onion root tips and distilled water.
Preparations of onion root tips slide.
First, cut off 2 root tips for 1 to 2 centimeter long. By using dropper, put a small volume of acetic alcohol into the wash glass. Then, take out the root tips into the acetic alcohol, then leave for 10 minutes. While waiting for the root tip in the acetic alcohol, prepare the preheated 1 molar HCl. First, check the temperature in the water bath at 60 degrees Celsius. By using dropper, put a small volume of HCl into the beaker. Then, put inside the water bath. If the beaker are not stable, you may use tongs to stabilize the position of the beaker. While waiting for the root tip in the acetic alcohol for 10 minutes, you can prepare ice cold water in the beaker to let it cool. Put in the ice beaker and then let it cool. After 10 minutes, wash root tips in the ice cold water and then leave it for 4 to 5 minutes. After 5 minutes, dry them on filter paper. Alright, from the filter paper, transfer the root tips into 1 molar HCl inside the beaker and leave for 5 minutes.
Caution, root tips will be very fragile after heating. Please handle with care. Repeat the steps of washing in ice cold water.
Okay, after that, transfer the root tips into the clean slide. Cut the root tips about 1 mm long.
Make sure you cut at the correct end of the dividing tips. The root tips usually have a denser white color and appear more rounded than the cut ends near the onion bulb.
And discard the rest. Make sure you cut from the growing tip and not the opposite end. Tease the root tip with mounted needle so you can get a very thin layer of the specimen.
Teasing the root tips must be done correctly. This is to ensure the cells will be spreading out a little. The problem is, if you are being too gentle, the sample will be too thick and you cannot observe the behavior of chromosome even though you get the mitotic stages. But if you use too much pressure, the cells might break open and chromosomes can be distributed out randomly outside the cells.
Then add a small volume of acetylocene into the slide and leave for 2 minutes for staining. Okay, then put the cover slip at 45-degree angle and then carefully let it down.
Cover slip must be put at around 45-degree angle. This is to ensure there is no air bubble. Cover slip should be flat on top of the specimen. If the specimen is too thick, the cover slip cannot be put properly. It means you're not going to be able to observe mitosis.
Okay, and then blot friendly with several layers of tissue paper and press gently to spread the root tips.
Press gently cover slip by using strong vertical pressure using your thumb. This is to squash the tissue to make it into a single layer. Do not twist the slide or moving the cover slip from side to side. Cover slip might crack. The cells might be overlapping. The purpose of using these chemicals, for example, acetic alcohol, is to be used as fixative to stabilize the structures and to prevent chemical and structural changes while the cell undergoes staining and mounting. HCl is to break the pectins that held plant cells together. Meristem tissues can be separated into individual cells. So this is to facilitate the stain to penetrate the tissue. Acetylocene is used to stain the chromosome with dark red color and also to stop the process of mitosis.
Then put your slide on the stage and then observe under 40 times objective lens.
Make sure to observe slide on low power first to scan the area which dividing cells are located. Then only observe under the next higher objective lens, which is 10 times, and then only you proceed to 40 times objective lens. You cannot directly observe under 40 times objective lens. The steps cannot be skipped. Locate the meristematic zone. Cells should be small, square-shaped with a large nuclei. If you get a rectangle cells, that will not be the correct cells. Results and observation. In the circle area, you can observe anaphase stages. So these are examples of several different stages that you can observe under microscope. If you do not manage to get a good quality slide, you may observe the permanent prepared slide as a guide.